make sure we're recording, which we are, and turn it over to Dr. Jeff Durr, who is the director of Hampton Roads AREC. Thank you both Jeff and Alejandro for putting together a presentation for us today. We look very much forward to it.
Thank you, Matthew. And I should start off by thanking Matthew Chappell for putting together this AREC seminar series. I think it's just a great idea and a great opportunity for people to learn about our various research stations. So again, I'm Jeff Durr. I'm the lead scientist here at the station, and I'm also the station director. I'm going to give you hopefully about a 15-minute overview of our station, our facility, our faculty, and I'll turn it over to Dr. Del Pozo, the featured researcher today for our research station. You've all seen this map just to show you where we are. We're on the bottom right, the Hampton Roads AREC, clear on the opposite end of the state from Blacksburg. We're roughly a rough triangle. You can see it outlined in red. We're bordered by Diamond Springs Road, Northampton Boulevard, and Bayside Road. That's our address for our research station. Just to give you a little perspective of where we're located, we're 1 mile from the Chesapeake Bay, approximately 11 miles from the Atlantic Ocean. So obviously, the two of those bodies of water have a big impact on our weather. And actually, we're now zone 8B. We had been in 8A, and we've been moved to 8B with climate change and things warming up. So we'll be quite different from the rest of the state, and we can grow some things here that you probably won't see anywhere else in Virginia at the other stations. We're very close to the Norfolk line. Virginia Beach butts up right up against Norfolk. We're short distance off of Interstate 64. We're on the way to the Chesapeake Bay Bridge Tunnel. If you've come to from Virginia onto the Eastern Shore, you went right past our station. We're about 300 miles from Blacksburg, and very valuable property where we're located here in Virginia Beach. So what do we work on? What's our vision? What's our mission? We want to be recognized nationally for our work in horticultural crop production, and I'll get into a little more about what those crops are, and environmentally sustainable landscape management. We are, our faculty all have an extension appointment, and almost all of us have a research appointment to work with the horticulture industry in Virginia. And that is predominantly the nursery industry, the greenhouse production industry, maintenance of landscape ornamentals and turf grass, small fruit production. And we are blessed with having the ability to cover all three major pest disciplines, insects, diseases, and weeds. And we have a stormwater management specialist here working predominantly in urban environments. He has worked with the nursery industry as well. A little bit more about our station. Total, we're about 70 acres for our property. About 50 acres of that is devoted to container and field research. If you're familiar with the nursery industry, a lot of that has switched to container, above ground container production. So we have container pads that try to match that production system. One thing that sets us apart, I think, from other ARECs is our demonstration gardens. We have about 10 acres, annual flower trials, the All-American selections, a variety of perennial gardens. We have a woody arboretum, et cetera. And I'll have a slide to mention some of those gardens. And those gardens are open year round, 7 days a week. People can come anytime. So if you're down this way on the weekend or during the week, feel free to stop by and check out our demonstration gardens. And about 10 acres for buildings, parking areas, and whatnot. We have 7 labs. The one that we're in right now that we're broadcasting from is our largest classroom. It's on the first floor. We have 2 smaller classrooms upstairs. We are blessed in having 2 graduate student houses on the property. So students can either come here, say in the summertime, to do research, or they may complete their classes on campus and then come down here for a year or 2 years to actually do their research. we have now in total 8 greenhouses. We just completed our newest greenhouse, and we will be starting on another greenhouse, putting up a new greenhouse shortly. So if we gave this talk again next year at this time, that number will go up to 9 greenhouses. Just a little bit of history on our AREC. There was a vegetable company, the Southern Produce Company, that was composed of truck crop growers, predominantly vegetable growers in the area. They felt there was a need for research on vegetable crop production in the area. 1907, they purchased 59 acres. Back then, Virginia Beach was just a small town at the ocean front. The rest of it was Princess Anne County, and then later on, there was a merger of Princess Anne County and Virginia Beach. So if you look at area, Virginia Beach is a pretty large area because it was predominantly a county in the past. In 1920, that produce company set up a 99-year renewable lease with the state for it to be a research station. You can see Virginia Truck Experimental Station. So that was established as a state agency in 1920. So we have passed our 100-year anniversary. We're now on our, I guess you could say, starting our second 100 years. Back then, this was a big vegetable growing area. So that was the focus. The acreage increased to 107 acres, but then it was later reduced to our present 70 acres after some land sales. With the growth of the population, housing in our area, the growth of the nursery industry, research activities were kind of redirected in 1967 to address the need of the nursery landscape industries. And if you look back on the history of our station, we've gone under different names over the years. Then it was changed to the Virginia Truck and Ornamentals Research Station. And for those of you not familiar with the term truck crops, those are crops that are grown and basically trucked to market. And again, that was predominantly used for vegetable crops, but we feel our nursery crops are also trucked to landscapers, to garden centers and whatnot. But that was, at that point in time, the station was a research station with the state, but separate from Virginia Tech. That changed in 1985 when General Assembly established the Virginia Truck and Ornamental Research Station as part of Virginia Agricultural Experiment Station. That's the research arm of the College of Agriculture and Life Sciences at Virginia Tech. So 1985, our station, as well as our sister station on the Eastern Shore in Painter, became part of the Virginia Tech system. We were renamed in 1994 to the Hampton Roads Agricultural Research and Extension Center, and that's our current name. Southern end of the building was renovated in 2003 and connects to the 1950 wing of the building. And one thing that's interesting, Virginia Tech does not own the land that we're on for our research station. Most of the land is owned by Virginia Beach. We have a pond we irrigate out of, about an acre or so. Norfolk owns the pond and an easement around the pond. The state owns a portion where we have our demonstration gardens. Dominion Power has actually an easement also across our station. And we're on something you never hear of anymore. We're under a 99-year renewable lease, so we're only a few years into our new 99-year lease. Just to give you an idea, this is not a complete list, but some of the stakeholders, groups that we work with Virginia Turfgrass Council, the Tidewater, the Turfgrass Water Conservation Alliance has funded some of our projects. Virginia Nursery and Landscape Association. IR4 project is a federally funded project to support the development of pest control products for high value crops. American Hort represents the nursery greenhouse industries across the country. Work with the North American Strawberry Growers Association, Virginia Strawberry Growers Association, Virginia Greenhouse Growers Association, and you see Virginia Turfgrass Foundation has funded our work in the past. And again, that's not a complete list. That's just giving an idea of some of the groups that we work with. We have an extensive Master Gardener program here. We have over 100 Master Gardeners that volunteer And without their help, we wouldn't have the gardens that we have at our station. We do a whole host of tours, classes, student tours, tours for garden clubs, various groups that come out here to see the research we're doing and to visit especially the display gardens. We do field days here at the station, various workshops over the year, and I'll talk about a few of those, pesticide applicator training. And we have over 40 groups that utilize our facility, either our large classroom, the outdoor facilities, or combination of the both. So we work with a lot of different groups here at the station. Our major classroom gets a lot of use. So if you want to hold an event here, make sure you contact us well in advance, because it does get used heavily. Just to give you examples, some of the events that we've had this past year. We hosted back in January the Come to the Bay Turfgrass Short Course. We will be hosting that again in January 2025. So if you have anybody, if yourself or others interested in commercial turfgrass management, put that date down to come in January this coming year. Our Master Gardeners had a spring plant sale back in May. We hosted the Mid-Atlantic chapter of the International Society of Arboriculture on May 11th. They got to use some of our trees for some of the demos that they put on. A berry tour June 4th. My Turfgrass Field Day was June 27th this past year. We also hosted the Virginia Nursery Landscape Association Field Day back in July. Our Master Gardeners, along with our assistants, put on the Fall Gardening Festival in September. Again, we host school tours, elementary schools, all the way up to community college, everything in between, groups that come out to take advantage of our research and demonstration areas. Something new for us, we assisted Mark Ryder and his team for the International Soils Conference, and we had a cover crop demo here at the station, as well as digging a soil pit that the attendees got to walk down through. And we host every year Tidewater Community College horticulture classes. And that's just some of the events key for us each year. Go through the faculty we have. We have 8 faculty, counting our newest horticulturist that will be starting in January, our nursery production specialist. Again, we covered the 3 pest disciplines. We have a plant pathologist, entomologist, and myself as a weed scientist. We'll have 4 horticulturists, a water engineer, we have a research scientist, and Dr. Schultz is our retired entomologist, former station director. 9 staff, we have administrative assistant, mechanic, farm manager, and assistant to the farm manager, social media specialist, and 4 technicians. We advise postdocs, PhD, masters, and OMAL students here at the station. Plus, we hire wage employees. We like hiring tech students, but they tend to go back in mid-August. So we like Virginia Wesleyan students. If you're not familiar with it, Virginia Wesleyan is just up the street, 5 minutes away. And we've hired a number of those students for hourly wage positions, and a number of them have gone on to be graduate students in our program. So that's been a really nice connection that we've had. I'll give you our faculty. Again, I'm the weed scientist here at the station. Dr. Fox is a horticulture associate looking at landscape sustainability. You'll hear more from Dr. Del Pozo, our entomologist. Dr. David Sample is, I call a water engineer looking at stormwater management. Our newest faculty member will be starting in January. He's moving in December to Virginia Beach, is Dr. Chris Cione, and he's filling a need that we've had. That position, nursery production specialist, has been vacant for about 4 years. Really looking forward to Chris coming on board, continuing that program. Dr. Hong is our plant pathologist. Dr. Eric Stolnick has been here about a year. He's our greenhouse production specialist. And Dr. J.S. Santani is our small fruit specialist here at the station. Just real quick, some of the things going on here and types of research and extension programs. Dr. Fox works closely with our master gardeners, overseeing the display gardens. You can see a list, and that's not a complete list of all the different gardens that we have. A neat tree trail, buffer garden. You can go on down the list. woody arboretum, various perennial gardens. Again, over 100 master gardeners help us. I should say master gardeners and some other volunteers work with us on the gardens. We couldn't have the gardens that we have without the number of volunteers that assist us. There are some pollinator plantings as part of our demonstration areas near Diamond Springs Road, but we also have a research pollinator garden that Dr. Del Pozo and I have been utilizing. Just some real quickly, some of the programs. A lot of this slide is work with David Sample, and Larry Fox works with David closely in a lot of projects. Urban stormwater management is his specialty. He does a lot of monitoring and modeling work, selection, placement of best management plans. Lori focusing on sustainable landscape practices. Dr. Sample has completed a lot of projects across the state, Fredericksburg, Richmond, Virginia Beach, etc. One of his newer projects is looking at soil erosion, modeling what's coming off of solar panel sites. Working Sea Grant, monitoring contaminants is another newer research effort of Dr. Sample. We've had floating wetlands work from the two of them, and Dr. Fox has a rain garden here that she utilizes for teaching purposes. Dr. Samtani, again, our small fruit specialist, focusing on strawberries and blackberries, cultivar evaluation, fertility programs. He's done a lot of work with his students on methyl bromide alternatives, primarily being anaerobic soil disinfestation. So you can check with him on that type of research. He has a high tunnel to work in his small fruit program. Dr. Eric Stolnick and I have received a funded project from USDA that'll be actually funding our second new greenhouse. So we'll be, we've started that process to assemble that greenhouse. He just completed a hydroponic lettuce trial. He's looking at greenhouse lighting systems and photoselective shade materials. Dr. Hong, I'm sure you've heard about his work on boxwood blight, a devastating disease on a major nursery plant species and a major component of landscapes in historic sites of Virginia. Previously, he did a lot of work on Phytophthora and irrigation water, how to manage that at container nurseries, identifying the species and how to manage them. I won't go into detail on Dr. Del Pozo's program. I've listed some of the key nursery ornamental insect pests that he is focused on. My work, I do weed control work in turf grass, nursery production, landscape maintenance. There's some of the weed species, annual broadleaf weeds key in container production. We do yellow nutsedge work every year, and we've expanded the last few years with work on Kalinga, some of our key broadleaf weeds in turf, buttonweed, violets. We've done a fair amount of work how to selectively control one grass out of another, to take Bermuda grass or Dallas grass out of, say, tall fescue. And we do some work in non-crop species, and we've done a lot on Japanese stilt grass or microstegium. I also oversee the turf program here at the station. We have about an acre of tall fescue, over 2 acres of hybrid Bermuda grass, about a quarter acre of zoysia, a quarter acre of St. Augustine. We work with the NTEP program, which is the National Turf Grass Evaluation Program, and we've had trials, variety trials in tall fescue, zoysia, St. Augustine, as well as low maintenance, warm season, cool season turf grasses. We have we call it a rainout shelter, where we do drought tolerance work, evaluating turf cultivars for their ability to tolerate drought and come back from a drought period. And we have a very large drone. We can spray herbicides with that drone. We can apply granular materials. That drone can lift over 100 pounds. So we plan to do a lot more drone research here as it relates to herbicide application. And we also have a number of sensors that we can do additional physiology work with our drone. So just kind of, to wrap things up. For my part, we want to be nationally recognized for our work, addressing especially horticulture production, landscape, turf management. Again, integrated pest management is a key part of what we do. That crosses over into horticulture production and water resource management. Again, the focus area is nursery, greenhouse, small fruit, turf grass, ornamental maintenance in our programs here, research and extension programs. We do have a quarterly newsletter, and I thought I'm going to end my talk with this slide. You can click on that QR code if you want to subscribe to our newsletter. We'll have another issue coming out in December, which we've been real pleased with. Our social media person, Elena Martin, has been a big help in kind of getting the word out on what we do. And one of those big efforts was the newsletter. With that, second move forward. Maybe I'll hold off until the end if we have any questions, and I'm going to turn it over to Dr. Del Pozo. Let's see.
We can stop share. Just put it like, you can close that one.
Yeah.
And it should be.
It should be right there. All right, you want to take it
Yeah.
from there?
Yeah, I can.
And that's what you want to use.
Okay. Good afternoon, everybody. My name is Alejandro Del Pozo. I am honored to speak before you. I just want to make sure that I can share my screen with you guys so you can see. Here we go. Perfect. So you're gonna get what I call an executive summary of the breadth of the topics, the research topics, and it's also extension as well within the entomology program here at the Hampton Roads Research Station. So I titled this as Expanding the Toolbox, Applied Research for Improving IPM in the Green Industry. I'm going to start putting in front, like right the first thing, the acknowledgement. Dr. Dare emphasized, but I also want to emphasize how crucial our collaborators, cooperators, and funders for our work, all the way from NIFA to the growers. You can see all that, and the specific collaborators as well, or personnel that we have, and you can see those names in this slide. And the people, that's what matters the most. We can be all professional. That's our conference on the left, and we can be as happy in our natural environment in the field, setting up a trial in New York on the right. I thank them because everything you'll see today is part of the hard work of the entire team here at the entomology lab at the Hampton Roads. So without them, I need to praise them and say thank you from the get-go, because nothing that you're gonna see is gonna be possible. So I want to center the topic what the entomology lab is doing for the past 4 years, and it's what I call the Applied Insect Ecology Lab. And what we're trying to do is to propose this insect pest management tactics. It is for the green industry, ornamentals, and turfgrass. The major topics within this lab are biology and ecology of insect pests, control tactics, and also all the alternatives to broad spectrum insecticides, including new chemicals or even new technologies like drones. I love this diagram because this is what centers what we're trying to provide to science-based solutions to growers across Virginia and beyond Virginia and the region and across the United States. So IPM as a core has the research and extension, but it's more complicated. So I love this diagram because it illustrates how complicated and convoluted integrated pest management can be. So based with that, keep that in mind. So how can we achieve successful suggestions for growers that are in need of those science-based solutions? So I decided to take upon one of the components, not the traditional components, but one of the components. What about the knowledge and resources that we have for improving the IPM programs for, in this case, nursery, ornamentals, and turf? So we can go from monitoring, winter biology, all these different topics that I am really interested in, and we're dedicating time, effort, and research in the lab. But here's the very first question that is the overarching topic. So we all read from books when we go to study that a diverse system has to be, it's sustainable, it's resilient, higher diversity, reduces the actually presence of pests and promotes beneficials, and there is an equilibrium. So now this is a beautiful picture. There's like 6, 7 different tree types in that shot. Why is it in ornamentals pests are a problem? So we are intrinsically, we have hundreds of taxa. Why is that we have key pests that actually cost money to manage? So if you think about it, we have annual plants and we have multi-year plants. So we are sustainable through time. We have multiple, we're diverse. But what is happening? So why the pests are present in the system? So as I told you before, I can definitely devote my entire career to look for one pest at a time, and I will not run out of pests. And I don't think that's the goal of this lab. So what I did is, as soon as I got hired, I went back to the growers and said, can you tell me what are the top important pests? Those are what you're seeing right now. So across the state in Virginia, we have issues with red-headed flea beetle, Japanese maple scales, ambrosia beetles, and now the new invasive box tree moth. So I'm going to present some of the summaries of some highlights and some research that it's already published and ongoing, and to address those top priorities that the growers get the feedback for this entomology program. So the red-headed flea beetle, you see it on the top right. It's a chrysomelid. It's a defoliator. The adult is the problem. On the bottom right, you see a highly defoliated hydrangea leaves, and basically growers are spraying up to twice a week because of this insect. So it's costing a lot of money, and the growers were loud and clear even during my interview. I still remember vividly that during my interview, it's like, Alejandro, what are you going to do about red-headed flea beetle? So we jump at it right away. So one of the things, because we didn't know anything about it, besides it is present in the systems from a long time ago. We don't understand why is it a problem, and why is it a problem now with no information. So the very first key, and as what I was trying to explain to you, is that we're really interested in this natural history, phenology, and biology of the pest, because that will be the foundation to actually understand what can we do next. So in this case, my first grad student, Ellie Lane, helped me to get together all the phenology of this. And so you can see the difference for the adult activity through the year for this insect as an adult phase. And we were able to recognize, and we were able to tell the growers they're expecting to have 3 peaks of activity throughout the season. And since weather conditions will change, those peaks are gonna slightly change as well, as you can see, but they're always gonna expecting those 3 peaks. So that's where I recommend and suggesting growers to always pay attention to those peaks. Then questions around, well, when can we scout? When can we spray? If we're spraying twice a week, is there any specific activity within the day? So we documented the activity through the season, but what about within the day? Is there something that's going to change? So we were able to see that really, really active. These adults are really active from 11 in the morning to that will be 3 p.m. and then they're sedentary for late afternoon all the way to early in the morning. Why is it important? Because if you're early in the morning and you go and scout for something and they're not going to be visible to you, and you can underestimate a hotspot or something like that. And on the opposite side, if you know that you have a hotspot, you wanted it to, right after your lunch break, you can just go and check those hotspots and double check if those are actually having issues with this red-headed flea beetle. So this has been informing growers in terms of, if I don't have enough time to do scouting, when exactly I can go to in order to find them actively fleeing around and see them and corroborate if there is a problem or not. So that was red-headed flea beetle. I'll move to the next one. If you remember that graph with the three figures. So within that second portion, we have the Japanese maple scales. These scales are, they have the waxy cover. They're really tough. They're really difficult to manage. And we have on the bottom right, that is a boxwood plant heavily defoliated because of a heavy infestation of Japanese maple scale. So you can see that potential there. Growers are also spraying in several applications through the season. So this is something that needed to be addressed. So again, the book says a lot of things, and up till someone takes a look at it. So the book said there is only 2 generations, which we proven that it's not holding up in Virginia. And then the second is that this developmental activity is stopped during the winter time. Our research showed that that is actually not holding true. And the idea is that we were able to document, that's the figure that you see, that we went back into the nurseries and we collected samples in January, February, and March. And then we were able to characterize what was in those samples. You can see how the bars, the number of specimens start moving from crawlers, early matures in January, all the way to immatures, a little bit of adults in February, and then you got adults and immatures in March. What does it mean? So growers have this idea about everything is hibernating, everything is overwintering there, so I shouldn't care about it, and everything's going to start in March, April. Well, the news is now that things are progressing and you have multiple stages within your trees. So therefore, if you're going to do an insect growth regulator that only affects immatures, that might not be something to only rely on, because you're going to have adults, so you need a different product or a combination of products to address both the adults and the immature. So that was something that now the growers are using in order to inform the decisions. Once they getting into the spring, they know that these insects are still developing, so they can scout, determine what is the ratios, and then spray it properly. The next one is ambrosial beetle. This is an insect that my predecessor started working with this and the growers, and it is still a problem. And it will remain because their biology is so phenomenal. I'm in love with this guy. You can see here what happens when you have attacks for ambrosial beetles. We have multiple species. You see in the top 2. In this case, Xylosandros cruciscus on the bottom is the one that is the predominant one in the entire Virginia. Those noodle-like toothpicks are the characteristic damage within those trees. So the tree on the right, if you go to Home Depot and Lowe's, and you're gonna see that, you're not gonna buy it. And that tree is $150 to $200 a piece. So you are actually, if you multiply that, you are looking at a lot of loss. And I don't blame them. This guy, as I told you before, these guys are so phenomenal and amazing. You know, they love ethanol. Who doesn't? I mean, I do. I love ethanol, you know, and what happened is ethanol is produced when plants are stressed physiologically. And that ethanol is emitted through the stress plant. So the plant is weakened, and then ambrosial beetles queue on that ethanol. And then in order to find that weakened and stressed tree where they can put, because they don't feed on the tree, they bring a fungus that they can put it. So they farm that fungus inside the tree. So I call them that the Airbnbs for ambrosial beetles are actually the trees. The problem is physically, you're going to see those toothpicks and you're not going to buy it. So on the left, this is something in Louisiana that happened. So that is a can of gasoline that had a high concentration of ethanol. And the person who published that paper is, you see those round black dots, those are the rear end of the females going through the plastic in order to get inside the can, because that was gasoline with ethanol. On the right, there's pictures from when you're enjoying yourself in your deck and you have a glass of wine or the beer, and all of a sudden this black dot lands on it. Well, that is an ambrosial beetle that thought that your beer was a stress tree. And that's basically what you're going to encounter. So now, I dare you, how many ambrosial beetles are you going to count from now? Now you know that those are the insects that are flying into your beer or a glass of wine. I like to say that this is one of the most expensive graphs that I generated in my entire career, thanks to my predecessor, Dr. Pete Schultz. We have over multiple years of data, and this is something that has been helping the growers to understand when successfully time the applications for this beetle. So we use the bottle that you see on the right. That's the most high-tech trap for insects that it's ever been developed. It's a 2-liter soda bottle that it actually connects with a solution for collecting and you have the ethanol pouch inside the bottle, which mimics the tree, so that the ambrosial beetles are going to get inside the trap and they're going to land into the killing agent on the second bottle on the bottom. And then we are able to count and see what the activity. So this graph is over 20 years of data in our eastern part of Virginia, and summarizes all that, where we can, with with some credentials, we can say, hey, you're gonna have a problem starting the first week of April all the way through the years. And I know this temperature is important, all the different variables are important, but at least we can have an expectation when to start sampling, when to start doing some timing, and the power comes from the multiple years that's been this sampling program's been done. I personally I'm interested in the landscape ecology, so I'm using Ambrosio virus to describe the landscape ecology of a pest. So on the left, the first graph on the left, you see those are interpolations. We have this GPS marks grids that we're trying to understand how Ambrosio virus is moving through the system, coming from the woodlots, how far, how long. So that gets actually paired with fly meals, where we want to understand the fly potential. So in the middle, that's the video. We glue Ambrosio beetles, teeny tiny beetles, into these arms, and we actually calculated the distance based on the number of rotations that they go through in this equipment called the flight mill. So far, we were able to document that up to the average is around 100 meters, give or take 300 feet. And that helps the grower understand how far this Ambrosio beetles can fly. On the right, you see this is a project that one of my PhD students is leading. This project is about marking with fluorescent powder the individuals that we capture alive and then re-release them in their own natural space. So we're not moving or removing things. It's whatever's in a specific environment, we mark them and we then trace them down in order to understand how far they can fly. So we have so far we have recorded 225 meters away from the release points. We're able to collect fluorescent powder mark Ambrosio beetles adults from these trials. The next one, the last one, is box tree moth. A picture says 1,000 words. So the two on the left, that is July 15 for 2022. That's a boxwood, a healthy boxwood. And next to it, on the right of that first picture, you see 14 days later, that same boxwood, it's gone. That's how this caterpillar is its potential. So on the top right is the caterpillar, pretty distinct hairs and lines and the head capsule. And then on the bottom, you have the beautiful adult that it's the one that is going to be the one that moves around in landscape. So again, with our expertise, we're able to bring to the picture how to document the natural history, biology, ecology, and also the phenology. In this case, on the top, you see in 2 years of data where we are able to document 2 peaks of activity for adults. On the middle bars is the small caterpillars. We'll still have 2 peaks of of caterpillars. And then on the bottom is the late instars, so the big caterpillars and the activity through the year. And this project has been done in New York. We also have more about, we're collecting with all that data, we are trying to construct and validate growing degree days and base 50 Fahrenheit in order to predict when the activity of these caterpillars should start in the systems. So we are able to do some, and you can see on the left, we are throwing some calculations, so we're trying now to validate it with our own data in order to see if this growing degree day models can inform growers when they have a problem with these caterpillars. On the right, you see a regression line where we were unfortunately able to document that even though predators and parasitoids are present in the system, they're not using box tree moths as a prey item. So potentially at some point this is going to regulate itself, but so far 2 years of data shows that the predators and parasitoids already present, they're not actually affecting the box tree moth caterpillars in any way. All right, so that's the knowledge for the pests and biology, life history. So what about, how can we do with that? So I like to look into tools and technology. So you're going to see a brief summary of tools, control tactics that my lab it's trying to promote and conduct research in order to share that information with interest growers. So the first one, it's a automated trap that actually take pictures, send it to the cloud, recognize the insect that is being captured, and they send you an alert to your phone. So that is an existing technology that some growers are using it, but it needs more dissemination. My lab tested it, and there's no difference between a regular trap and automated trap. So this is a really ongoing thing, and I'm excited to start seeing this type of technologies. On the picture on the right, those green squares is actually the computer recognizing the adults for this. In this case, it's Diamondback moth. They recognize it, counted, and send you an alert because the count from the day before gets subtracted for the new count, and can give you the number of adults captured per day. Then these are my good friends, the beneficials, and people always forget about them. There is a sweet array of beneficials that are working for us, for the growers, and I'd like to promote this. I'd like to remind the growers that these are our allies. So you can see from predatory mites, ladybugs on the top, like wasps in the middle, lacewings on the bottom, hoverflies, minute bugs on the top left. There's all this sweet array of beneficials that are working for us, right? So here's an example. How can we use drones to do augmented biological control? This is Kevin Hill, still my collaborator in California, and the company is Bug. So Kevin is loading a drone in order to release lacewings into a field, and that is being mixed with rice hulls, and those rice hulls contain the lacewing eggs. And then he plans the mission for the drone, knows exactly where it's going to go, and then you just start, and you start going to see how those rice hulls with the lacewings are going to start dropping from the sky, literally dropping from the sky, in order to be placed into the field. And this is not testing. This is happening as we speak. Actually, I was crazy enough to team up with. I'm going to do a little shout out with Tim Bryan, our newest entomologist. Crazy enough, like me, in order to test now this in cotton. So he's interested in looking if augmented biological control can be performed with drones and cotton, which is a little crazy. People don't like that idea, but crazy is good. Another technology, it's mating disruption. So we're looking for alternatives to boost and strengthen IPM, right? You saw the drones releasing beneficials. Now you see a technique which is mating disruption, using sex pheromones in order to confuse males. So on the left, you see this diagram. They're trying to explain how mating disruption works. So you have these dispensers that actually emitting the pheromone of the moth, and that overwhelm the system. So the males are not able to navigate it, find those females, copulate, mate, and actually don't have progeny. So the idea is that you do mating disruption, you don't have caterpillars, and you don't have damage, right? And we did this in Western New York. On the right, you see in one person from the USDA handing one of the dispensers. There's the solid bands that have impregnated the pheromone for the box tree moth, and we pair that to 2 applications of Bacillus thuringiensis, which is a bacteria really specific for LEPs that actually works into the digestion system of the caterpillars and kills them. Just to give you a brief overview of what we did, this is a table from 2023. We knocked 220 doors in order to enlist people in 3 different towns in New York in order to place these dispensers. So it was quite an effort, and that required a huge team, which I'm thankful that we have that team still with us. And actually we did this mating disruption this year, so we finished everything back in the last week of October. So we needed to, in order to organize ourselves, we had to have all these GPS maps on the left. The green means that those properties agreed to receive those placements of the dispensers. So we got to go in the people's yards and put the dispensers. The red was the one that they didn't want to do anything with us. The yellow was a maybe, and the brown, it's the controls that didn't receive any dispensers at all. Now, you saw those different technologies, but we still need to have answers about efficacy of insecticides. So my lab is very well equipped to run all these different bioassays in order to understand the efficacy of different insecticides at the laboratory, greenhouse, and open field settings. One example is one beautiful collaboration with my counterpart in Georgia. She met Joseph. We both were able to try new technologies for nurseries in order to see how can we manage red-headed flea beetles, in this case, the immature that lives in the pot. So we tested top dressing, which is placing the chemical on top of the pods. We tested drenching, and we tested multiple chemicals that most of the growers are interested to know. besides, trying to walk away from neonicotinoids, which are having some backlash. So some of the results is that there are specific products that are not going to work. So the ones that are on the right, those ones that are having a lot of adults coming from those spots, and the ones from the left and the ones on the right, you can see there are promising results where adults were suppressed and even defoliation was suppressed through those years. So we're really excited, and that's this new technology. Now, Alejandro, it's a lot of money that we're going to use, labor to drench or to do the top dress. Well, that is, at least we proved the concept that this targeting the pot, it's going to give some benefits. So now I think it's the next step will be how feasible or how new technologies can help us implement this changes. This is busy, but this is on purpose, because for ambrosial beetles, we're trying and trying. It has been trying, but unfortunately, the most successful solution is spray pyrethroids in order to protect the trees that are attacked by ambrosial beetles. So on the bottom, you see all these different names that you potentially you're not going to see it. But again, on purpose, I did this because I want you to understand that as a community of researchers, we're trying to find alternatives to pyrethroids, and unfortunately, nothing is actually working thus far. But the caveat is that now we have new technologies for adjuvants, penetrants, and other compounds that might help us to increase at least the longevity of pyrethroids, or maybe using a different chemical that is the second best in order to be used in the system for controlling ambrosial beetles. Now I'm going to move towards the technology. I'm part of KAIA, the Center for Advanced Innovation in Agriculture. Hopefully you guys are aware of KAIA. KAIA, it's a great group that we all share this passion about using technology to improve our system. So in this case, I'm using remote sensing in order to provide more information to the growers so they can make an informed decision, right? One of the things that we are doing is collecting imagery, light reflectance with drones. We have a drone in our lab that is a multispectrum, and we also have a hyperspectral radiometer that it's a portable that we can collect all these different light reflectance. That being said, with those collection of data, we can calculate all these different, what is called indexes. They're gonna, in a way, be a proxy for plant health or other stressors. In this case, normalized difference vegetative index is one of the indices that has been used most commonly, the NDVI, right? So you can see that we can collect those wavelengths, we can translate that into a more, I call it amenable, but in a more relatable number, which in this case is the index, the NDVI index. So here's an example. We fly this is one of my collaborators in Central Virginia. These are boxwoods in the field. We fly the farm with our drone. We're able to calculate NDVI, and then we can tell the grower, hey, by the way, these are the parameters that we believe these are healthy plants, and these are plants that might be a stress. So would you like to take a look at that, since everything is at the GPS detail? So we have all the GPS, that grower can be able to go back and ground truth these observations. And so far, there is some correlations about irrigation and maybe some pruning that was able to be picked up by this NDVI. So we are still working on this as we speak. When was it? Yesterday, the team collected NDVI from another nursery, and we're trying to understand what are those relationships with stressors. It could be insects or something else, and the NDVI, so that will be the light reflectance. This fun project in turfgrass, I'm also using turfgrass as another platform to deploy all these new technologies. So in turfgrass, we looked at using remote sensing to characterize areas that are refuge for pollinators and beneficials. So we were able to fly the drone, collect the reflectance, and trying to come up with the index that tells what areas have this specific indices that can be potentially correlated with vegetation that promotes the presence of pollinators and beneficials. So we run a bunch of different, actually we're 13 different indices, and we're able to find that CIPI and PSRI were one of the two. Doesn't mean anything right now to you, but those are specific indices that tends to pick up orange and yellow, which are the flowering plants that will be present in a specific corners on a golf course. So everything was done in a golf course in order for the superintendents to keep those protected areas with no mowing and promote the presence of beneficials and pollinators. So this was a really neat project, and we are gonna test and validate this some sort of indices in the coming years. All right. Again, I told you that I was honored to give you this some sort of a executive summary of the major research areas in the entomology lab that I'm leading here at the Hampton Roads Research Station. So in terms of my applied research and extension philosophy and components, I like to use the biology and ecology of the insects, and specifically the target, in order to inform those decisions that are going to implement IPM programs. Combining pest control tactics, it's still the cornerstone. So we're validating what is existing, and we bring in new technologies in order to expand that toolbox that we need. The research is always applied, so I like to go to the farms, visit with the growers. We do have something that I call the IPM schools every year, where I visit every collaborator that I have to their facilities, and I present the data that we collect from their own farms, and we're able to have like an hour discussion about what could be the plans for next year. So that's been really successful, really well taken by the industry, and we're going to continue to do that. So because we are an applied lab, we go to the farms and set up and execute research projects with the farms. Within research of the areas of research that I'm promoting, biology and monitoring, cultural and biological control, and chemical control in order to avoid insecticide resistance. So I always conduct an informal need assessments in order to guide my program, not only for the research, but also for the extension. And I'm always available to provide the technical support. Some of the growers have my cell phone number, they call me, and that's what we're supposed to be present for them and provide that support, that technical support. Just to round it up about the specific technical information I present, here in Virginia, we have key pests, which is the red-headed flea beetle, Japanese maple scale, and ambrosial beetle still is a problem. Box tree moth hopefully is not going to come, but it's likely that it's going to come. It is already in 5 different states right now, and they're all quarantines. And I'm happy to report that the data that we generated here at Virginia Tech helped USDA APHIS to get to the compliance agreement that we have at the nationwide. So the industry is not impacted. So the sales are not a stop anymore. So if you sign up your compliance agreement, you will be able to sell outside of the quarantine. And this is a huge effort, and all lead by USDA APHIS PPQ. And we were able to support that effort as well. Biological control is something that can be implemented now with new technologies, like, for example, the video you saw, Kevin Hill and flying the drone. Augmented biological control can be something that now it can be feasible and economically affordable. There's new technologies for remote sensing, mating disruption, and even documented areas that could be refuge for pollinators and predators. Those could be new tools or tools that can improve any IPM program. And again, this IPM program actually, aids to reduce both the environmental and the economical risk. So with the pillars having the chemical control plus the cultural control, and trying to find those alternatives as well. That's my house. I wish that could be my house, but I like to end this to give you a flavor of what the green industry is. It's what it brings to us as a community. That peaceful, and I want to go home. I want to spend time in my garden. I want to spend time in my lawn. I want to run barefooted with my kids. So that's what our impact it's all about. It's to promote that societal benefit, and we are the support to the technical support for those growers to be successful and have those more of these pictures everywhere. With that, that's all I have. I hope I didn't run out of time.
No, you didn't. Thank you so much.
Perfect. Yeah, I don't have my phone with me. You go to the chat. There's questions in the chat. Okay.
So my computer malfunctioned halfway through. So if you guys have questions in the chat, please do read them off on your end.
Nope, no questions, comments.
All right. Does anyone have any questions?
I would say they'll come and visit us. We're fun to work with. Jayesh, yes, we have some questions here in the room.
Yeah, thanks for the great seminar. The pests that you highlighted, you were like focusing on the phenology. So that's something like they have not been explored before.
For the case of red-headed flea beetle, we didn't have an idea. Like we were blind in terms of there's nothing. Actually, the paper that we published is the first paper that actually suggested that, formally that we have. It was anecdotal, the number of, and Pete can corroborate this, everybody was talking about it, and there was nothing formal and standardized being done. So the first thing we did is do multiple locations, multiple years, and trying to formalize that, and that's what we did. And that informs when or how you're gonna deploy those control tactics.
So this was like not done at the national level.
No, we are right now as a part of a collaborative group, and we just submitted an SCRI, so a specialty crop block research initiative grant, where we're going to look at the national level, because this is a problem in Florida, Georgia, South Carolina, North Carolina, Maryland. Mostly this region is the one that is having this issue with the red-headed flea beetle.
George Davis has a question. Can you discuss some of the cost processing software in identifying the adult diamondback moth?
Yeah, so
No, that's not my question. That's not my question.
Oh, no, that's not George.
Can you discuss some of the cost differences or comparisons in terms of just eradication, no other environmental anything, just for the consumer who wants to have a yard that looks like this? Can you talk about some of the different cost differences across these different pets?
Okay, if I understand correctly, and the budgets they're going to be needing and the costs they're going to be needing, of course, I present this picture. That's our ideal, which it's going to drive most of the pest management tactics that we're going to implement. So budget wise and cost is going to be a variant in my understanding about how much would you care about a specific end products and how much you're willing to pay. Unfortunately, I don't think I'm equipped to give you specific numbers or budgets, but I will be able to say that this gradient would depend on how much of the new technologies are implemented, what you will use for alternatives, if you have a specific insecticides or pesticides that don't want to be used. So there are multiple factors that are going to be part of this budgeting and cost that's going to be ranging from affordable to really expensive. And what are the means for that? I think that's a great idea for a future project where we can compare economical costs for, hey, I would like to have a 100% organic managed lawn, or like that picture compared to I don't need neonics at all, but we can use something else. And so you see that kind of will be my point. And I'm not sure. I'm not aware if there is something like that being done at this level yet.
Let me just simplify the question. Which of these pests, you covered four, I believe, which of these pests you think are more difficult to control?
The ones that I presented?
Yeah, of the ones you presented.
All of them are difficult to control and really expensive. That's why I decided to study them.
Okay. All right. Thank you.
Yep.
Sorry about that. I didn't have my glasses on, so I was reading one question and another one popped up. But from the Winchester gang, what is the accuracy of your image processing software in identifying the adult diamondback moth?
Okay, so that is done for a third party. I know Villas at Eastern Shore, they were trying to do something about taking pictures and identify, say, beetles that are on plants. I know those are efforts happening. The remote trap that I present is owned by a company called TrapView, and TrapView charge per trap, and they provide because they have thousands of images. So their libraries are from Europe, China, Israel, and they brought it to North America. So this company has this beautiful library, so that they're able to process. So I did a study comparing the accuracy, and the automated trap in terms of identifying was 98% accurate.
That's pretty good.
The caveat though is that you can program your trap to give you alerts every hour, all the way to every day. So I don't know if I were you, but if I'm a grower and receiving alerts that every hour there is a new insect, I'll go nuts. So that is something that's the difference, right? The traditional is the traps is we check them every week, once a week. This trap gives you the resolution of insects every hour. So I don't know. So those are things that our industry we're not that used to yet. Is that meaningful? Is it something that we need? And every hour, every day, so every week it's been working. So that's something that we need to address in the short term period.
Do they have like a threshold for like so to like advise you if there's like
you can, yeah. So there will be threshold. So the company also sells you in some sort of like a website based software that you can set up your own thresholds, and then based on those thresholds, you're gonna get a different number. So you do definitely get an alert when the insects are there, but that doesn't mean anything, right? So then you can set up your own thing with historical data and all that, and say 3 in a day, it's a problem. Boom, and they'll send you the additional alert. So yes, that is possible. Pete.
Hot fucking night, you've got also, at least by my porch light, there's all sorts of things flying around. This the ability to differentiate.
Yes. Yes. So the trickiest part with that 2%, what we were able, and I remember this, when we were able to see is that there was this odd placements on the sticky liner that the computer couldn't recognize. But most of the, let's say it went really flat, right? But as long as you have something and the computer recognize it, because it compares that, so it recognizes the insect, it compares with thousands of images, you know, it's an AI, and then says, all right, so this is the insect I'm looking for.
Okay. Do we have any other questions? Oh, a couple popped up. Number one, can we access today's recording? Yes. So there is a link that Suzanne Pruitt had put earlier in the chat. I will send this out to everyone as well in an email, but there's a VAES website in which you can assess the recordings from all the seminar series. You can also look up the Tidewater Ag Research and Extension Center YouTube page, where you can find all the recordings and more on other topic matter. So we will get that to you. Again, the link is earlier in the chat. You can scroll up and find that as well. Go down. There was one other question. Enrique, do you feel end users are being educated to help properly identify and treat to help minimize resistance to ensure longevity of current effective pesticides?
That's a great question, Enrique. Thank you. And we are the stewards to promote and preserve what our tools, our current tools are actually, you know, in place. We're trying our best. We are equipped in my lab to do dose response and sets of mortalities and do all this dilution. So we try our best to, if we have a hint or an assumption, or a grower says, Alejandro, I'm spraying, I'm not getting results, that hints us to say, maybe there's something going on with insecticide resistance. So I think it's our responsibility as researchers to do that. We are equipped to do that. And we also are presenting this at every grower organized meeting that we can. So education is key, and talking about it and having the data to show that. So far, for example, we are looking at a couple beetles, and we're going to analyze the data and see how we stand. So this is an ongoing process where monitoring for insecticide resistance is key.
Fantastic. Well, we're a little bit past time. Thank you everyone for attending. I do want to remind everyone we do not have an AREC seminar for December. We will join back in January 31st, where Eastern Shore AREC, Dr. Mark Ryder and his staff and faculty members will be presenting a little bit about that station. So again, thanks everyone for attending. We hope you have a great holiday season, and we'll talk to you later.
Thank you. Thank you, everybody. Take care.